Unpredicted that Andres couldn't make it, unfortunately, so I'm speaking instead of the scheduled time of Friday. We just learned about this a few minutes ago, so Kirk drove me to my hotel and I had to jump out of the car, get my laptop, run. So I know how Tom Cruise feels like in Mission Impossible. Okay. So I also apologize. I don't have a kind of a presentation that's more kind of tailored towards the specifics of what people might be interested in. So what I'll give is instead my promotion seminar, which was a couple of weeks ago. So it kind of gives a very broad overview of everything that's going on in my lab, and doesn't give any information on any other computational analysis we're doing. I apologize. Okay, so let's start with kind of what we're working on. So we're working on kind of analysis of the microbiome together with the host genome and information. We're basically trying to understand what are the host genes that kind of interact with the microbiome, which genes in the human host or other hosts can affect the microbiome, and vice versa. How does the microbiome can affect host gene regulation, for example. So just a couple of slides to kind of begin about the human microbiome. So I'm sure everybody has heard about the human microbiome. There were already a couple of talks on the human microbiome, but just very, very briefly. So there are 100 trillion organisms in and on the human body, which is 10 times more microbial cells than human cells. The number 10 times is not really accurate. It could be, some papers say it might be twice as many. Some papers say it's about the same number. Another paper said it basically depends on the last time you ate a big meal. So the number doesn't matter. It's just there are a lot of microbes, and there are thousands of different bacterial species in and on the human body. There are 8 million different bacterial genes, which when you consider kind of from the human genetics perspective, we have about 20, 30,000 genes. So this is a few orders of magnitude, a couple of orders of magnitude more than the number of human genes. You can kind of just think about the kind of the functional potential of these genes and what they can do in addition to what our own genes can do. They're found anywhere on our body, any kind of mucosal surface. They're found in the skin, in the mouth. The largest number of microbes is found in the GI tract. And then another tidbit I like is that the weight of all these microbes, to put them together, is 2.5 pounds, which is about the weight of the human brain. A joke that I like to make is that it's always a number I keep in mind when I step on the scale, just to make sure. You can just take that off. Okay, so another question that's pretty important is, why is the microbiome important? Why do we care about the microbiome? What does the microbiome do? And the microbiome is actually involved in a lot of different functions. I'll kind of try to highlight some of them throughout my talk, but just very briefly, the microbiome is involved in metabolism. So bacteria can metabolize all kinds of foods that we cannot. They can provide us with all kinds of nutrients. They can interact with our immune system, and it can train our immune system, especially kind of in early age. And then they can also provide protection against infections and pathogens. And looking at kind of in the last few years, maybe the last 5 to 10 years of microbiome research, there were lots and lots of studies that have shown that the microbiome is correlated, or changes in the microbiome are correlated with all kinds of disease states of the human host. This is just a kind of an outline where you can see there are changes in microbiome in asthma, in hypertension, in all kinds of neurological disorders sometimes, inflammatory bowel disease, obesity, a lot of different diseases, metabolic disorders, a lot of different diseases where you see changes in the microbiome between people that are healthy and people who are sick. And this is really interesting for me as someone with a population genetics background. If you consider this slide, which you've seen yesterday, I think, where you see all the different genes and all the different loci in the human genome that have been associated with complex traits and diseases, you can see that a lot of these diseases are actually the same diseases. There are also linked to changes in the microbiome, things like digestive system disorder, cardiovascular disease, metabolic disease, immune system disorders. They're all linked to the microbiome as well as to host genetics. And basically what I'm saying is you have a disease and you have a change in the microbiome that could be maybe affecting the disease or could be just affected by the disease. We're not sure the directionality there in this arrow in the middle. And we also know, obviously, that from 20 years of GWAS studies and more than that, looking at human genetics, is our own genes affect, obviously, disease complex diseases in humans. What we're interested in looking at is this direction. So how does host genetics, how does human genetic variation affect the microbiome and interact with the microbiome? Can our own genes, basically, our own genetic variation affect the composition of the microbiome, the abundance of different microbes that are found in and on our body? And I'll basically tell you about two kind of stories today where we looked at the effect of host genetics on the microbiome. One, just looking at common genetic variation, so genetic variation that segregates in humans and how that is correlated with the microbiome, but also looking at disease-causing genetic variation. Specifically, we're looking at somatic mutations in cancer and how variation in these mutational profiles in colorectal cancer tumors can affect variation in the microbiome. And then on the other side, we have environment. As a population genetics, I'm looking at the microbiome kind of as a quantitative trait, and I can look at variation in the microbiome and separate it to variation that's due to host genetics and environment rather than everything else. And I kind of mentioned a few stories. I'm not going to have time to get into them, but I kind of mentioned a few stories, basically a couple of papers that we published recently. One looking at dietary effects on the microbiome, looking at other flora in the microbiome and how they affect the microbiome, specifically parasites in the gut. And then another really interesting study they published looking at transmission via social interaction in a non-human primate model. And then the last thing I'll tell you about is how does the microbiome affect us in terms of disease? I'll kind of try to wrap it all together and tell you about a new model that we're using. We're trying to look at genetic or variation within the microbiome and see how that variation affects gene regulation in the host using a new model that we're developing. Okay, so starting with this, just looking at host genetics and the effect of the microbiome, there were a few studies that basically tried to look at the effect of host genetics on the microbiome. The initial studies didn't actually have any human genetic data, but they just look at relatedness, which is here on the x-axis, and kind of look at the correlation between relatedness and the similarity in the microbiome, which is on the y-axis there, and you can see there is a correlation that may or may not be significant, which basically says that maybe more similar, more related, closely related individuals have more similar microbiomes. There are a few other studies since then. This is by now like almost 10 years old, a study where there were also included some twins in the study where they found that monozygotic twins have more similar microbiomes compared to dizygotic twins, although it wasn't significant. But one thing they found is that individuals within the same family have more similar microbiomes compared to unrelated individuals. And there were a few studies looking at a really nice model of mouse. This is a figure from Benson et al., where they actually looked at basically try to map sites in the host genome, in this case in the mouse, and try to correlate that with the microbiome. And they used the QTL mapping approach here. And they actually found a number of loci in the mouse genome, which are those colored, those bands here. Which you can see each of these loci is basically correlated with variation in the microbiome with the same color, with the kind of the tree, the phylogenetic tree of the microbiome is like plotted in the middle there, which is a really nice approach to look at this variation. Some of the challenges here is that it's hard to really map the specific gene, the specific sites within each of these very big loci.
Yeah, what is the circle? This is the genome.
So the circle is the mouse genome. Yes, and each of these bands is basically a locus that they found using QTL mapping. So, where there is a association, a correlation with the microbiome.
How it's related to the tree of life?
So this is the tree of the microbes that they had in the study, and you know all the microbiomes, and basically the color of the different branches in the tree is basically linked to the same color of the band. So you can see that this kind of basically variation, or this area in the mouse genome is correlated with that part of the microbiome. Yeah. Okay, and this is another recent study where there was actually genetic variation data where this is Julia Goodrich, who was back then was a grad student with Ruth Lay and Andy Clark at Cornell, and what Julia did here is actually use a pretty large data set from the Twins UK cohort, where we had monozygotic and dizygotic twins. And you can basically look at the variation or the change of similarity in the abundance of different microbial taxa and compare that in monozygotic and dizygotic twins. And you can actually calculate that way the heritability of each basically taxon within the microbiome. And again, you have the phylogenetic tree. Doesn't look exactly the same as before, but you can see the phylogenetic tree of the microbiome, and each of these colors basically represents the p-value on the right hand, and on the left hand side you have the basically the heritability of this specific taxon, which you can calculate using this approach. So as you can see, most of the microbiome is not really heritable, so it's not really affected by host genetics, seems like. But some taxa are heritable and actually have a significant heritability based on this analysis. Okay, so taking kind of all these studies together as kind of the starting point to what we're doing, it seems like there is an effect of host genetics on the microbiome, but it's really hard to quantify it, and it's really hard to pinpoint the specific genes and loci that are associated. Okay, so what we wanted to do with this project is basically look at the effect of host genotype on the microbiome. So we want to see which genes and which pathways are involved. We want to understand the mechanism of interaction. We want to see the role also in controlling disease. And the approach is we're trying to combine host genetic data and metagenomic data, and we're using association techniques. And the data set we're using is data from the Human Microbiome Project. And another kind of goal that we had is only use publicly available data. So just one slide in the Human Microbiome Project. This is the data that we're going to use for this project. So the Human Microbiome Project was a very large NIH-funded project with the goal of characterizing the microbiome across different body sites in the human body. So they had 15 different body sites. They're aggregated into these 4 what they call super sites, and you can see the airways, basically samples from the nose. The oral sites are 9 different sites that are sampled within the oral cavity. 4 sites on the skin, then GI tract, as usual, is represented by fecal samples. And basically they use shotgun sequencing to explore the different microbial genomes. And then they use 16S ribosomal RNA sequencing to characterize microbial diversity. The difference between these two approaches is that 16S ribosomal RNA sequencing basically focuses on one small region within the ribosomal RNA gene, and sequencing just that region in order to have enough information on genetic variation in that region, or genetic diversity in that region, to be able to map each read hypothetically to a single taxon within the microbiome, while shotgun metagenomics is basically sequencing all the DNA in a sample and getting information on all the DNA in your sample, and then taking that DNA and be able to kind of reconstruct information on the microbiome from those reads. Okay, so this is a really great project, tons of data, really broad. One issue that we had when we wanted to start our project is actually no human genetic data that was available as part of the Human Microbiome Project. So it didn't do any sequencing or genotyping. Since we started this project, this has been done, and last year there was a paper published looking at SNP genotyping information, whole genome sequencing information from the Human Microbiome Project individuals. But when we started this project, and by the time we published this, which was three years ago or so, there was no, this data was not available. So what do we do? Well, there's a known issue in metagenomic shotgun sequencing, which is called a host contamination, when you look at human data. When you sequence all the DNA in a sample, some of the reads are going to come from the host. So usually in microbiome data analysis, these reads are discarded as contamination. But what we wanted to do here is see if we can take those contamination reads and say something about the host. When we take those reads and map them to the host genome, actually learn something about genetic variation in the host. So we're very happy with it. That actually worked. It actually allowed us to, just from this contamination data, to generate whole genome sequence for the HMP individuals at about 10x depth. The reason we could do that, and the reason you can't really do that in almost all metagenomic shotgun sequencing studies, is that we can take data from across the different body sites, and we could aggregate those contamination reads for each individual. So that actually allows us to get millions and millions of reads for each individual that come from the host genome, just from this, using this approach. If you think about kind of traditional microbiome sequencing studies, so you do shotgun metagenomic sequencing, for example, in fecal samples, you're not going to have enough, even close to enough data to actually do something like that. So it was a very unique approach that we can apply here. Okay, so we have this host genetic data with microbiome data. The first thing we wanted to do is just look at kind of overall correlation. And one thing we find is if you plot the identity by state, or kind of the similarity between the host genomes, the human genomes on the x-axis, and the similarity in the microbiome, which is 1 minus the beta diversity, which is another way to just look at basically similarity between two microbiomes, we see a correlation. It's not a very strong correlation, but it's significant. So each dot here, again, is just a pair of individuals, and you have the similarity in their microbiomes, and their genomes seem to be correlated, which basically means there is, so individuals that have more similar genomes also have a more similar microbiome. And we find the same pattern. This is just looking at the fecal samples, where we find the same pattern looking at the microbiomes of 10 out of the other 15 body cells. Okay, and in addition to kind of looking at this overall correlation, we also applied a model where we looked at across the basically the genome and focusing on SNPs that we identified in our data set that are found within coding regions of the genome. We can actually find SNPs where we have genetic variation in the host that's correlated with the microbiome. And this is another kind of our way to kind of try to reproduce the same plot that you saw previously for the mouse, where you see the host genome, in this case the human genome, on the left-hand side, and the microbiome here, and you can actually find, we can actually identify sites here where you see a correlation between genetic variation and the microbiome, and some of the genes are also listed on this plot. So there are a few kind of interesting results that we found here. I think the most exciting probably result, the most interesting correlation that we found is this one. So we're looking here at the abundance of bifidobacterium in the gut. And basically used the abundance of bifidobacterium as a quantitative trait, and we basically did a genome association study where we tried to look at genetic variation across the genome and the correlation of that with the abundance of that microbe. And what we found is that there's a hit basically on chromosome 2. You see here on kind of the left in the red dots, and that hit was significant after genome-wide FDR correction. And what we found is that this hit is, if you zoom in, it actually overlaps. It stands right on top of the LCT region within the human genome. So this is really interesting because the microbe, so bifidobacterium, which is the microbe that we studied here, is really interesting because it's a genus of bacteria that metabolizes lactose. And there were some studies that have shown that bifidobacterium sometimes actually prefers lactose over glucose. And the LCT gene, where we find the association, is the gene that encodes the lactase enzyme, which is the enzyme that kind of facilitates the digestion of lactose. So basically, the fact what we found is the genetic variation here is correlated with the level of bifidobacterium. It kind of makes a lot of sense. And there were a few kind of studies that validated that. Actually, all the other studies that came out since our study that also had human genetic variation in the microbiome, they all kind of found the same association. And this is a nice kind of paper that kind of described this finding, and there's this really nice illustration that kind of explains it. So basically, if an individual is lactase persistent, so they have an active lactase enzyme which will break down lactose, so they have a low abundance of bifido. Bifido is marked with these red microbes there. So if they have an active lactase enzyme, which is on the left-hand side, regardless of the diet, they're going to have a low abundance of bifidobacterium because there's not going to be enough lactose. But on the right-hand side, you see the case where someone is lactase non-persistent. And in that case, if that person also consumes lactose in their diet, the bifidobacterium will be able to use it and metabolize and then grow. So this is the case where we find basically a correlation between genetic variation and the microbiome, which might be driven by basically genetic variation affecting the behavior and the diet of the host, which then affects the gut microbiome.
So is lactase non-persistent when they're lactose intolerant, or?
So lactase persistent are cases where individuals where you have the lactase enzyme active into adulthood. So these people can metabolize lactose. And non-persistent means they're individuals who can't metabolize lactose on their own. And in this case, if these individuals do, so the top row kind of indicates milk drinking or kind of any kind of lactose dietary component, and at the bottom, I think that indicates tea without milk, probably. So if they do consume milk and they don't have an active lactase, then the bifidobacteria will be able to use it. Yeah. Any other questions on this? Okay. All right. And when we take kind of, yeah, sure.
So for the lactase non-persistence, was that the dimension of lactase non-persistence, or was that based on the genotype?
That's all based on the genotype. That initial finding based on genotype. So there are a few studies now going on where they're actually doing kind of the lactose challenge test, where they drink like a gallon of milk and then measure glucose in the blood to see if actually lactate is actually active, and it breaks down the lactose or not. So there are more. It's more. I think it's more complicated than that. It's kind of a very simple illustration, but there are cases where you have the genotype. You know, people have the genotype or don't have the genotype, but can still consume lactose, and there's no real explanation yet that I know of why that happens. So this could be one explanation. Yeah. All right. All right. So another thing we found is that when we look at kind of the all the SNPs that we found correlated with the microbiome, when you do an enrichment analysis, we found a pretty strong enrichment of SNPs that are within genes that are related to cancer, and specifically colon cancer. And this kind of can use that as kind of a segue to the next project, or next project I'll tell you about. Sorry, probably skip that. Okay, just to make sure I'm on time. So this is a kind of a really interesting problem. It's kind of a mention a little bit of some of the computational analysis we're doing. So since we published this project in 2015, there's more and more interest in doing this kind of analysis of using the microbiome as a quantitative trait and try to use basically an approach sort of like a GWAS to identify SNPs in the host genome that are correlated with the microbiome. So to kind of address that, we developed a new method we called hominid that uses lasso regression to identify host SNPs that are correlated with the microbiome, and then basically use stability selection to identify for each SNP that's correlated with the microbiome the specific taxa within the microbiome that seem to be driving the correlation. And this was the work of Josh Lynch, who was a former grad student in my lab. And I don't have any other slides on that, unfortunately, but the software and code are all available on our GitHub. And then we also developed a visualization tool where after you run hominid and you have a list of these microbe gene associations, you can upload that into the visualization tool, and then you can actually see, you can visualize this using kind of a network visualization approach where you can kind of add genes, reduce genes, change the cutoffs, and then basically change the visualization approach. Yeah.
How you measure the correlation between a certain SNP and in the microbiome and the human genome?
Right. Yeah. So there are a few ways to do it. So the microbiome, this is what I was actually going to say now. So the microbiome is a really interesting thing to look at as a quantitative trait because it's composed of a large number of potential features or potential traits that you can use. So each microbe can be considered a trait, and then you can also think about if you aggregate the microbiome data, depending on what type of data you have functional data, you can actually look at the abundance of different genes in the microbiome, the abundance of different functional pathways in the microbiome. You can look at actual SNPs in the microbiome. So it's very broad, and it's a really interesting problem too because it's hard to define the the quantitative trait you want to look at, and there's also a lot of basically intercorrelation. So all the data is usually abundance data, which is relative abundance. So if one microbe goes up, by definition everything else goes down. So it's all intercorrelated. And if you want to use something like, people have used something like, just looking at the principal components of the microbiome, for example, to try to do the mapping. That usually doesn't work when in a kind of a GWAS approach because the effect of each SNP is not that large that you would see it on a PCA of the microbiome. So that's why the lasso approach works a little bit better because it actually identifies those kind of correlations you wouldn't see. You wouldn't see just using like principal components of the microbiome. But yeah, one of the main problems, like define your traits and figure out how to normalize it so it would fit, into the specific model you're running for the association. There's also a problem of just the data being very sparse. So a lot of the times you have microbes that are found in one individual, two individuals, and nowhere else. And maybe that microbe is really strongly correlated with genetics, but you wouldn't be able to find it. The sample size are relatively lower than what you have in human genetic, like in GWAS studies, where you have millions now. Here, the largest GWAS that I've seen of the microbiome is in the order of like 10,000, I think.
10,000
individuals. Yeah.
Where?
I definitely. So there was a paper on. So there were a few studies that were smaller.
Maybe 16x.
Yes. Oh, for sure. Yeah. It's all 16s. Yeah. So 16s is much cheaper, so you can actually do that. Cheaper to do the sequencing, not to collect the sample. But yeah. Yeah. Any other questions on this? Yeah.
So how do you normalize the data
when you're looking at associations and here?
So it really depends on your the model you're running. So there are a few different ways to do it. When we ran kind of the model, first of all there's a step of filtering to kind of remove a lot of the microbes and reduce the multiple test burden. So what we did in our analysis, just to reduce the number of microbes we're actually looking at, to make sure we only include ones that are more prevalent in our data set. And then the normalization is really so we used a number of approaches. The one I don't remember. So we used the box-Cox normalization, which is a simple normalization for the initial analysis, and in current analysis we're using a different normal. But yeah.
So the thing that takes into consideration the fact is compositional data, and then you're basically in a simplex and not. I mean.
So the normalization is we're running normalization at each taxon. So this is happening only when we run each taxon as a trait in the model. So in that case, yeah. But if you want to run the entire microbiome, this is a whole different problem. That I'm not really sure how to do that. I mean, like I said, there were a few studies that just use the principal components of the microbiome, which has other problems. Yeah.
So, in your also regression, so are you do the reversing of the normal G1? You are trying to put the SNPs as the outcome and
Yes.
use different quantitative trait of microbes.
The microbes, yeah. Yeah, so basically what we're doing is, it might be not the exact approach that people are using in the GWAS study. We're basically looking at the SNPs as the outcomes. So each SNP is basically the outcome, and then the microbes are the different features in the model, yeah. Right. All right, I don't have a lot of time at all. 15 minutes, yeah, I like finished 20% of my talk. Okay, so what I'll do is I'll skip a lot of stuff and I'll talk about one project. So just still looking at genetic variation. So I'll skip a bunch of these. These are really interesting, I'm sorry. Okay, so I'll just focus on one study that was actually published just a couple of weeks ago, looking at disease-related genetic variation and the association between cancer mutations and the microbiome. Okay, so remember this slide. So a lot of the sites that we found in healthy individuals seem to be sites that are involved in genes that are involved in cancer and colon cancer specifically. So this is a really important problem. Colon cancer is one of the most common, one of the most deadly cancers. So about 100,000 new cases a year, 50,000 deaths a year. So it's a very important problem to study. And there's an emerging link between colon cancer and the gut microbiome. There were a lot of studies to try to basically look at the effect of cancer on the microbiome. So some of the findings are that there's a decreased microbial diversity. So people with colon cancer usually have a lower diversity in the microbiome, so fewer taxa within the microbiome compared to healthy people. There's an altered abundance of a number of bacteria that seem to be recurring in a number of these studies. Fusobacterium is one of them, and specifically Fusobacterium was also identified in some other studies as a possible causal role. Where a bunch of studies actually had cancer cell lines, and then they treated them with Fusobacterium and found changes that might be related to cancer development and cancer progression within the cell lines. The microbiome seemed to also have an effect on the efficacy of chemotherapy and immunotherapy, where you can actually sometimes predict the success rate of chemotherapy and immunotherapy just based on the microbiome, the gut microbiome. The gut microbiome, specifically for colon cancer, can actually also be used as a screening tool, whereby you have the normal screening tools for colon cancer, which is either detecting blood in stool or colonoscopy. So if you take the traditional kind of non-invasive approach, just detecting blood in stool, and you add the microbiome to it, you actually increase the accuracy of this approach. And then obviously the microbiome is linked to a number of risk factors for colon cancer. So diet is linked to both colon cancer and microbiome. Inflammation is also the same. Okay, so there's really no kind of strong, even considering all of this, there's really no strong consensus of what is the cancer-related microbiome, and the mechanism of interaction is not really clear. For my approach, again, as someone working on host genetics and the microbiome, is really interesting because cancer is a genetic disease. Cancer is a disease of, caused by mutations to the host genome. So basically, studying this interaction and actually looking at the cancer microbiome and considering the mutational information from tumors might be informative and actually tell us more about the role of the microbiome in causing cancer and the effect of the microbiome on the disease. Okay. So traditional studies where they try to look at the colon cancer and healthy individuals and look at the microbiome usually use a simple approach where they have a cohort, kind of a case control design where you have people with colon cancer, people without colon cancer. And as usual, you look at their fecal samples. This is the kind of a non-invasive approach, relatively easy to get. And usually what they do is 16S ribosomal RNA sequencing, which is just, like I said, looking at the one region within the microbial genomes that can tell you about the taxonomic composition of the microbiome. Some of the challenges with this approach is there could be a lot of environmental factors that could be biasing the study. So if, let's say, the two groups had a different diet, that might have an effect. If the two groups have had different exposures, life histories, and so on, that could also have an effect on the microbiome and on cancer development. Looking at stool samples might also not be ideal because colon cancer is kind of a localized disease, and looking at the microbiome that's found right on the tumor, right adjacent to the tumor, might also be relevant. And sometimes you don't see those signatures when you look at colon cancer. You see, or you look at stool samples, you don't see the same microbes there. And like I said, in my talking about the previous project, there are also host genetic factors that might be involved, and these studies are usually not including that information. So what we're doing is looking at only at colon cancer patients, and we're looking at their tumors and at a normal sample right next to the tumor, adjacent to the tumor. And that way, we can actually get kind of information on the change in the microbiome that's specific to the tumor, kind of correcting for a lot of these biases when you do case control design, because it's all corrected for basically the same individual. And at the same time, we're also taking a biopsy from the tumor and from the normal tissue right next to the tumor, and have information on the genetic mutations that are happening in the tumor in order to basically try to find associations between that and the microbiome. And this is a project that was kind of entirely headed by Michael Burns, who's a former postdoc in my lab, and a large number of collaborators and other students in my lab that I won't name now. Okay, so there are basically two questions we're trying to answer here. First, just looking at the microbiome in these samples. Is the microbiome that's attached to tumors different from that of the healthy colon from the same individual? Are there any changes in bacterial diversity? Different taxa, different functions might be enriched? And then can we use the microbiome to actually tell us something about the tumor or predict things like the tumor stage? And then once we incorporate genetic information into the analysis, can we actually look at the mutated genes within the tumor and see associations between that and the microbiome? Can we see the microbiome changes that's associated with specific genes and pathways that are mutated? Can that tell us something about the mechanism of interaction? And then can we use the microbiome to actually predict information about the tumor, about the tumor's mutational profiles? Okay. Just start by just looking at the microbiome data and looking at diversity. What we find is that there's an increase in diversity in the tumor biopsy compared to normal tissues. This is kind of maybe surprising because a lot of studies that looked at colon cancer actually found decreased diversity in fecal samples compared to normal samples of normal individuals. Here, again, we're looking at only at cancer patients. When we look at the diversity at the tumor compared to the diversity right next to the tumor, we find an increase in alpha diversity and other diversity measures. The advantage here is that we actually have matched samples. So from each individual, we can match the sample from the normal tissue to the tumor, and we can see whether there was an increase or decrease for each individual. And this is just looking at kind of those, each line basically connects the samples from the same individual in the normal tissue and the tumor biopsy, and the red ones are going up, the green ones are going down. This is just looking at the change, basically the tumor minus the normal, the change in diversity with a 95% confidence interval. So this kind of highlights the importance of maybe just looking at specifically at the tumor and not at fecal samples. Yes?
When you say the tissue from the same individual, how adjacent?
So it's pretty close. So it's less than 10 centimeters. So we made sure it's relatively close, as much as you can. Yeah.
Are they homogeneous?
Right. No. So there are different locations in the colon. So we tried to we ran a model to try to see if that has an effect on the microbiome, and we didn't find any significant effect of the location in the colon. But I agree that I think if we had maybe a larger sample size, we might have found something, because that kind of always comes up when you look at normal colons. Basically, there is a difference in the microbiome, but the fact that these are close together, I think, might indicate that this effect is local. Yeah, yeah.
So do you take more than one sample, both the normal and tumor, from each patient just to get an idea of what the natural variability for each patient?
Yes. No, we didn't have that for this study, but we have an ongoing study where we have this kind of data, where we have multiple samples, so basically multiple biopsies, multiple healthy biopsies from the same individual. But yeah, that'll be informative. Yeah. Right. So this is just the diversity. So the alpha diversity, and each dot is so each pair of dots is an individual, and you have the basically a line connecting the normal tissue diversity and the tumor diversity. So this is kind of to visualize the fact that most of them are going up, some of them are going down, most of them are going up, and overall, on average, there is an increase. So red is going up, green is going down. Yeah.
I saw that in two previous slides you use 16S for tumor and exome sequencing for tissue.
No, so we had both from both. So we had 16S in the normal and tumor to look at the microbiome, and exome sequencing in the normal tumor to look at the mutations. Yeah, this is just looking at 16S now. Yeah.
Is there a way to control the amount of DNA that you get from the normal tissue and the tumor for getting the system and sample? So the total amount of reads that you get in those samples is the same?
Right. This is a good question. We haven't done that here, but you can always add things like the amount of DNA, the amount of the number of reads to if you run a model. You can always add that as covariates. That could be a way to control for that. And we've done that in the previous analysis. I talked about of the kind of the association analysis. Yeah, that's a good point. We actually haven't looked at that here. We should. Yeah, good question. There's usually a correlation. Yeah, more reads means higher diversity for sure. Other questions? All right. Okay, so we see this increase in diversity. We can also look at changes in the taxonomic composition of the microbiome. This is a figure that Michael really likes. He calls this the Christmas tree plot. And what this shows is basically the difference in the kind of the composition of the entire microbiome when you have paired samples. You have the normal and tumor sample from the same individual. So each row here basically represents a single individual. And for each individual, you have kind of the change in abundance, not the actual abundance, but the change in abundance from the tumor to the normal sample. So on the left-hand side, you have the taxa. Each color is different taxon. So you have the taxa that go down in the tumor, and the right-hand side, you have the taxa that go up in the tumor for each individual. So right as you look at this, you can see some really nice patterns. Like on the left-hand side, you have more red and blue. So this basically means there's kind of a decrease in the Firmicutes and Bacteroidetes on average, kind of in general across samples. And another interesting thing you can see is on the right-hand side, in yellow is Fusobacterium. And what you can see is Fusobacterium seem to be increased quite a lot, but only in about maybe 10 to 15% of the samples. And this is kind of consistent with other studies that found Fusobacterium kind of enriched in colon cancer samples in tumors, but only in a relatively small number of them, so maybe around the same proportion. Okay, so the next thing we wanted to do is kind of look at the relationship between the microbial communities and the stage of the tumor. So the hypothesis here is that the composition of the microbiome can be affected by all kinds of anatomical, physiological changes that are happening at the tumor. Basically what you wanted to see if we can basically use the microbiome kind of as a predictor for the stage of the tumor. And what we did here is use linear discriminant analysis to identify the taxa in the microbiome that are associated with tumor stage, basically separating the tumors into low and high stage. So we would have enough numbers to kind of compare it to just these two categories. And then we combine those into a single value, which we call the risk index. And then we used, basically we used the risk index for classification, and then we used the leave one out approach, cross-validation, basically to estimate the accuracy of our approach. And what we find is just based on the microbiome, we can actually predict the stage of the tumor with 80% specificity and 78% accuracy. So it's not doing a very bad job at basically just from the microbiome predicting the stage of the tumor. And you can also go back and kind of see what are the microbes that are mostly associated with different categories. Here you have the microbes associated with high versus low stage. The next step, which I think the most interesting analysis, actually include the mutational information. So basically what we had here is whole exome sequencing from the tumor and the normals. And we used the normals kind of as a baseline. We identified all the mutations that are unique to the tumor for each individual. Again, using a normal as a baseline for that individual. And mutations, as usual, we can kind of classify them to, all mutations, non-silent mutations. We focus specifically on loss of function mutation in this analysis because of the idea that those mutations would have the largest effect, largest functional effect on tumor physiology again, and that might have a bigger effect maybe on the kind of the microenvironment and on the microbiome.
What is the x-axis?
So this is basically just the number of mutations that we had. So the x-axis is just the patients. We had 44 patients. And the y-axis is basically the number of mutations that we found. As you can see, there are some tumors that have a large number of mutations, looking at all mutations and non-silent mutations. This is kind of things you also see in other cancer data sets like TCGA, where you have hypermutated tumors versus tumors that have fewer mutations. We didn't look at the all mutations and non-silent mutations at all. We just focused on the loss of function mutation in this analysis because those I think would be more informative. And this is, as you can see, there are maybe a few dozen, maybe a few hundred per individual. This is whole exome sequencing. So this is just mutations that are found within the coding regions of the genome. Yeah. Okay, so the gene where we found kind of the largest number of mutations or number of individuals that had a loss of function mutation is APC. And APC is a very well-known driver, one of the major drivers of colorectal cancer. And what we wanted to see is, can we use the same approach that we just used to look at tumor stage in order to basically predict whether a tumor has a loss of function mutation in APC, again, just based on the microbiome. And using exactly the same approach, we found it actually works, so sort of. So with a 73% accuracy, 80% specificity, which is not great, but it's pretty interesting because you just use the microbiome data and the taxonomic composition data of the microbiome, we can actually say, sort of, whether a tumor has or doesn't have a loss of function mutation in the APC gene. Yep. Sorry?
The features.
So the features are the relative. So we did 16S. So it's all basically an OTU table which we used for the features for this analysis. Okay, and we can also then kind of drill down and look at the microbial attacks that are associated with, specifically with APC mutations. We found a number that have been associated with colon cancer before, like Feingoldia. An interesting result here is we found Christian Synalacia here that seem to be enriched in tumors that don't have an APC mutation, APC loss of function mutation. This is really interesting because in the studies that I've mentioned, so if you remember the Twins UK studies I mentioned earlier, this is the taxon that was kind of found as the most heritable taxon in the gut microbiome. And there are a few studies kind of reproduce that finding. So it's kind of interesting to see that here. In addition to APC, we tested the 10 most frequently mutated genes in our cohort, and we saw that out of these 10, we can actually find for the top 5 of them, we can get a significant prediction. Again, just based on the microbiome, we can, with this accuracy here, we can say whether a tumor has or doesn't have a loss of function mutation. Another thing we can do is look at the microbiome. We can look at interactions within the microbiome. By interaction, I mean statistical interaction. So basically correlation, strong correlation between individual taxa. And what we wanted to see is if there is any effect of the mutations, specific mutations on kind of the network of interactions within the microbiome. And what you can see here is basically on the left-hand side, you have the network for tumors that have an APC loss of function mutation versus all the other tumors on the right-hand side. And you can see that the nodes here are exactly the same positions, but the links are basically the correlations we found based on the tumor, based on the mutations in the APC in those tumors. So what you can see is that basically an APC mutation basically causes a very big change in the microbiome and changes in the kind of the correlation between the individual microbes. So you see Christian Synalacia is higher abundance in the tumors that don't have an APC loss of function mutation. We can also take the mutational data and aggregate that across pathways. So instead of saying whether a gene has or doesn't have a loss of function mutation, we can look at an entire pathway and say, does that pathway, any gene within that pathway, any gene here has a loss of function mutation? And we can, again, look for correlations with the microbiome. As you can see, there are already, there's a pretty strong correlation between the mutational information of pathways and the different taxa. And we can use the same kind of prediction approach, which actually works better when you look at different pathways. And we used here a database of specifically cancer-related pathways, and we found that for most of these, or out of the 23 pathways we tested, 21 of them, we can actually find a significant prediction. So again, from the microbiome, we can get a pretty good prediction of whether there is a loss of function mutation in a number of these pathways that are very important in cancer, like MAPK signaling pathway, Wnt signaling, P53 effectors, and so on. This is kind of a list of all the pathways that we found where we can predict from the microbiome, we can predict loss of function mutations. The accuracy is around 80%, I would say, for most of them.
One quick question.
Yep.
So those accuracy scores, the cross-validation accuracy?
Yep. So one thing we still haven't done is we haven't applied that to any other data set. We only had our data set, and we still only have our data set because there are no other data sets that have mutational information in tumors and microbiome. So if anyone has heard of a data set, we can actually apply this and try to kind of reproduce this approach. That'd be great. Okay. All right, and then we can also kind of take those interactions between the mutational pathways and the microbes and kind of produce a network, and this is just showing kind of the different interactions between the microbes that are affecting kind of or that are correlated with the mutational status in different pathways. You can see some pathways are correlated with a large number of different microbes, like P75 and TR-mediated signaling, while some other ones are only associated with a small number of microbes, like canonical Wnt signaling pathway is correlated with Christensen-Laitier and very few others. Okay, so just to kind of summarize this study, and I think I'll end here. So we found that the tumor microbiome is actually more diverse than normal tissues in our samples. We found an enrichment of a number of bacterial taxa, and we saw the microbiome can actually use to predict tumor stage. And then when we include mutational information using whole exome sequencing on these tumor normal samples, we saw that the overall microbiome composition seemed to be correlated with mutational landscape, specifically in the pathways, and then the mutations affect the microbiome interaction network, and then we can use a prediction approach to basically predict mutations in specific genes, 5 out of the 10 genes that we predicted, and 21 out of the 3 KEC pathways, and also some PID pathways that have shown significantly just predict the existence of a loss of function mutation in that pathway from the microbiome. And that's it. There's another, I think I'll end here and skip the last part, just because there's not enough time. Sorry, it's getting to the conclusion. Okay, there you go. So yeah, I want to thank everybody involved in these studies, especially Michael, who's was a postdoc in my lab, now with his own lab at Loyola, and Josh, Karen worked on kind of the analysis of the host microbiome genetics, and I have a lot of collaborators without whom none of this would have been possible. This is our funding, and thank you very much for your time. Happy to take more questions.
I have 2 questions. The
Yeah.
first one is, I'm sorry if I missed, but when you look at the mutations in the tumor, are those number of mutations you observe, they are when you compare it to the same person's healthy tissue, right?
Yes, yeah. So the way we use whole exome sequencing in the same individual twice, so in the normal tissue and the tumor, and you basically find the mutations that are specific to the tumor of that individual that are not found in normal samples. So it's kind of a way to normalize for.
We don't know how homogeneous those tumors in cells with their mutations, correct?
No, that's a good point. Yeah, yeah.
Second question, it's more like a discussion point that I'd like you to comment on.
Mhm.
I'm sure you're familiar with Aaron Segal's work.
Yeah.
They recently published a paper in which they very clearly demonstrated by analyzing 5 different papers that use microbiome data plus host
Mhm.
genotype data, that LCT is pretty much the only thing that sticks out as like everyone finds. But other than that, there is nothing to a certain degree that they can't find a single SNP in one study within the vicinity of 1 megabase pair of the host genome
Yeah.
that results in the same phylum.
Yeah. Yeah,
It clearly shows that these associations between host genome and the microbiome is it's still driven by the environment because LCT is, associated with the environmental input. So what do you think about this? Like, is there really, would you conclude it's the environment or the host, and where do you stand?
Yeah, so this is it's a very good point. I didn't get into all these discussions here, but there are more and more, like I said, since we published this study, there were quite a few more studies that did similar kind of analysis or try to identify SNPs that are correlated with the microbiome. There's not a huge concordance, like you said, between the studies. There are a number of reasons why that could happen. One, I think, is some of these studies are done in different populations, and different populations might have a different diet, different environmental effects. So the environment might be actually controlling the microbiome. So if you don't have a microbe there, you're not going to find an association with that microbe, right? So there's a kind of an environmental effect that's very strong. The environmental effect is much stronger than the genetic effect. That's for sure. I mean, we know that the microbiome changes immediately if you just eat a different meal, right? So I'm not surprised by these results. I think we need bigger cohorts, bigger kind of more standardized sampling and analysis and sequencing for these studies. I think we need something very important. We actually need to look at the same individual over time, which none of these studies have done. And in microbiome, something unlike other GWAS studies, like we look at like BMI or height. Height is not going to change dramatically, probably, but microbiome changes could be pretty dramatic over time. So we have a study that we're kind of just starting where we don't have genetic data, but we have relatedness information. We have lots of samples in a non-human wild population of baboons. So we have 20,000 samples of microbiome. What we're finding, I don't have it here because it's like very new results, but what we're finding is the heritability really depends on the kind of the time the samples were taken. So if you're sampling one season compared to the next, you can find different patterns of heritability. You find a very strong heritable microbe in one season, and then the next season it's gone. So I think looking at longitudinal samples in general would be helpful.
We have to leave the room now. So but feel free to ask the questions outside. Just take all our stuff. There's going to be another group that's